Hello friends, welcome back to Astral Biochemistry. Today we are going to deal the topic, the second part of carbohydrate chemistry, which is homopolysaccharides and heteropolysaccharides. So starting with homopolysaccharides, we have homopolysaccharides. The main homopolysaccharides that we are going to see are 7: starch, glycogen, cellulose, dextrin, dextran, chitin, first we will do inulin, inulin, and then chitin. So these are the homopolysaccharides. Usually the question asked is to differentiate between starch and glycogen. So we'll see starch first. So talking about starch, starch is a homopolysaccharide of glucose. It is a polymer of glucose. And it is the main storage form of carbohydrate in plants. So in the plant world, starch is the main storage form of carbohydrate. Now another point regarding starch is mainly composed of two polymers, which is two polymers. One is amylose and the other one is amylopectin. These two combine to form starch actually. So we'll see what are the points under amylose and amylopectin. So what are the amylose points? In amylose are amylose, the glucose units are linear in arrangement, so it is a linear polymer of glucose. Whereas this is a branched polymer of glucose. It is branched polymer of glucose. Now here, because it is linear, all the linkages will be alpha 1,4. Alpha 1,4 linkages will be seen here, alpha 1,4 linkages. But because there is branching here, you can see both alpha 1,4 and at the branching you will see alpha 1,6 linkages. Here it is alpha 1,4 only. This is alpha 1,4 and alpha 1,6 linkages can be seen. Now 20% of glycogen starch is composed by amylose, whereas 80%, most of the concentration inside starch is occupied by amylopectin. Starch. Another important thing is this is water soluble. This is water insoluble. So these are the main points in favor of starch. So when you discuss about starch, it is a polymer of glucose. It is a main carbohydrate storage form in plants. It's mostly composed of two polymers, which one is amylose and the amylopectin. Amylose is a linear polymer of glucose, so the linkages will be alpha 1,4 only. This is a branched polymer of glucose, so the linkages can be alpha 1,4 and alpha 1,6. 20% of starch is occupied by amylose, 80% of starch will be occupied by amylopectin. This is water soluble, amylopectin is water insoluble. So we'll move on to glycogen now. Okay, so the next thing that we are going to discuss is glycogen. Okay, next homopolysaccharide is glycogen. Now glycogen again is a homopolymer of glucose or polymer of glucose. Anyway, we are discussing homopolysaccharides only. So what do you mean by homopolysaccharide? That means all the units will be same. In heteropolysaccharide the units will be different. So glycogen is a homopolymer of glucose again. This and this, in this the linkages will be alpha 1,4 and alpha 1,6. Okay, because there is branching. So this is a highly branched. So you can maybe write polymer of glucose or a highly branched polymer of glucose. Okay, you can write it here, highly branched polymer of glucose. So because it is branched, it is highly branched, it is globular in nature, and it will be compact, and because it is tightly packed, and it can be tightly packed, so it occupies less space. Okay, occupies less space. So one important thing that I forgot to mention about glycogen is it is a main storage form of glucose in animals. Okay, in animals. So in plants it is starch, in animals the storage form is glycogen. So in animals it can be stored in two places, one in the liver as glycogen, liver glycogen, and also muscle glycogen. So these are the two areas where glycogen is stored here. Yes, so I guess that's about glycogen. Yes, so we'll move on to the next homopolysaccharide, which is, we have finished starch and glycogen. Next is cellulose. Okay, cellulose, sorry. Yes, we'll talk about cellulose now. Now cellulose again is a polymer of glucose, but the linkages are beta 1,4. Okay, linkages are beta 1,4 linkages present, and this is present in plants. Okay, this is present in plants mainly. Humans cannot digest this. So when you take a vegetable foods or which contain plants and all, the vegetarian food, when you eat, what happens is these humans cannot digest the cellulose contained in that plant part. So cannot digest the cellulose, cannot digest this, because what enzyme is not present in humans? We don't have cellulase. Okay, cellulase is an enzyme which can break this beta 1,4 linkage. Cows and other plant eating animals will have this enzyme, but our humans cannot have this, do not have this cellulase enzyme. That is why we cannot eat grass and all, right? And so we cannot break down this cellulose when we eat plants also. So that is why it is an important dietary fiber, important component of dietary fiber. So important component of dietary fiber. So because it is acting as a dietary fiber, it will increase the stool bulk, thereby relieving constipation. Okay, because when you have cellulose in your gut, cellulose will attract a lot of water into it because it is osmotically active. It will draw water from the surrounding intestinal cells and all, so the stool bulk will be more and it will relieve constipation in a way. Yes, and because it is a dietary fiber, it will also decrease the absorption of glucose and cholesterol. Okay, it will decrease the absorption of glucose and cholesterol. So when you have fatty food or carbohydrate rich food, if you have salads with that, if you eat some salads or have some plant parts with that, what happens? Cellulose will help to decrease the absorption of glucose and cholesterol. So what happens? This will help in the management of diabetes mellitus as well as hyperlipidemia. Right, because it will decrease the absorption of glucose and cholesterol. Hyperlipidemia, and also you can maybe mention obesity also. Okay, so diabetes mellitus, hyperlipidemia, obesity, all this can be kept in check because of using cellulose or increasing the dietary fiber in the food you consume. Okay, so these are the points about starch, glycogen and cellulose. Okay, what is the next? So in the 7 homopolysaccharides that we are going to discuss, the main 2 ones are starch and glycogen. Okay, questions have been asked more on those topics, but others can also be asked in the form of MCQs and all. Yes, so we have discussed 3. Now the 4th homopolysaccharide is, what is that? Dextrin. Okay, dextrin is nothing, it is again a polymer of glucose, but it is actually a partially digested product of starch. Okay, so when you have starch in your food, if starch containing your foods like tapioca or any plant parts, if you have as a food, the partially digestion will happen of the starch content and a partially digested product known as dextrin will be formed. Okay, don't confuse it with dextran. Both of them are homopolysaccharides. Okay, so this is a polymer of glucose. Again, the linkage is not so important. Just understand that dextrin is a partially digested product of starch. Okay, now about dextran. Okay, so 5th one is dextran. Now dextran, yes, dextran is a complex branched polymer of glucose. Okay, branched polymer of glucose. Now the uses of dextran are important actually. Uses of dextran. Okay, one will be as a plasma expander. Okay, plasma expander. You will see when you have emergency medicine posting or casualty posting in a hospital, in hypovolemic shock. Okay, it is given in hypovolemic shock, as the name indicates, because of blood loss or dehydration, usually due to blood loss, severe blood loss due to road traffic accident or something. If the patient loses a lot of blood, he or she will go into hypovolemic shock, and we can correct it with a plasma expander like dextran. Okay, so that is an example for that. Now it is also used as an anticoagulant. So these are the medical applications of dextran. And the uses, yes, another uses, it can be used as a lubricant in eye drops. Okay, lubricant in eye drops. And also another thing is it can be used in parenteral nutrition. Okay, parenteral means, enteral means through the gut. Parenteral means which is parallel to the gut. So outside through a tube, if you are giving parenteral nutrition, that is known as through the veins. If you are trying to nourish the patient, like some patients in the wards or casualty can actually come with very malnourished patients can be there, like those undergoing chemotherapy or during the last stages of cancer and all. Those patients will be undernourished. So to give nutrition to such patients, you can use parenteral route. Okay, enteral route maybe if they are suffering from esophageal cancer or metastasis of other cancers, those patients can't actually eat and swallow the foods. So such patients will use parenteral nutrition, which is giving the nutrition through your veins. Okay. So as a parenteral nutrition supplement also, we are using dextran. Okay. So these are the uses and the features of dextran. So don't confuse dextrin. Dextrin is a partially digested product of starch, whereas dextran is a complex branched polymer of glucose. Okay. Now this where did we? So after dextran we have inulin. Okay, which is the sixth one. Okay, inulin. Okay, inulin will be familiar to you from renal function test. Inulin is actually a polymer of fructose. Okay, all these polymers that we saw till now was polymers of glucose. This is a polymer of fructose. Polymer of fructose. Polymer of fructose. Okay. Now the linkage here present here is beta 2,1 linkage. Okay. Please remember a special linkage is there in inulin, which is beta 2,1 linkage, and this is a homopolysaccharide of fructose. Now what are the uses of inulin? Okay. As you know, to measure GFR, it is a better indicator, or to measure actually indicator for GFR other than over urea and creatinine. Okay, urea and creatinine. So this is a better indicator because it is fully filtered by the glomerulus, fully filtered at glomerulus, and it is neither secreted or reabsorbed. So this is a condition which matches the ideal substance that can be used for GFR. So when you compare with urea and creatinine, inulin will be a better indicator of GFR. So I am not going to details. These will be covered in renal function test, might have been covered already for you in the practical. So neither secreted nor reabsorbed. Okay. And also another feature of inulin is that just like cellulose, this can also act as a dietary fiber. Okay. Okay, polymer of one important feature that can be mentioned is polymer of fructose, which is found in inulin, is found in plants known as dahlia. Okay, inulin is present in dahlia. So the linkage is beta 2,1 linkage. See, the linkages that why I am stressing more is a lot of questions come from these linkage part, be it like simple monosaccharides or disaccharides that we discussed in the last class, or from homopolysaccharides like these. A lot of questions come. So these are the points about inulin. Okay. Yes. Now next we will discuss about the last homopolysaccharide. Okay, so the last homopolysaccharide is chitin. Okay, now chitin again will be not a new molecule for you. Chitin might be discussed already in your +2 classes when zoology was covered. So chitin is again a polysaccharide of, it is a polymer of not glucose, it is polymer of a special molecule known as N-acetyl glucosamine. Okay, N-acetyl glucosamine. So this is an amino derivative of glucose, or amine derivative of glucose. This is N-acetyl glucosamine. So many N-acetyl glucosamine, if kept like a polymer, this will become chitin. Okay. Yes. Now it is present in, chitin is present in the exoskeleton of insects. Okay, like if you observe some insects like cockroach or some other insects like that, it will have a outer covering, right? And those outer covering is known as exoskeleton. So such exoskeletons will have the main component as chitin. So insects, and also it is found in fungi, and yes, mainly fungi, and yeah, mainly in fungi. Okay, so these are the things in chitin. And the linkage, okay, before this we should have mentioned the linkage. The linkage is just like in cellulose, it is beta 1,4. Okay, beta 1,4 linkage. Okay, in inulin it was a polymer of fructose, but the linkage was beta 2,1. Okay, cellulose and chitin, it is beta 1,4. And all the other molecules I had mentioned already. Glycogen you have, which is a storage form of carbohydrate in animals. You have the linkage as alpha 1,4 linearly, and also branching will be present. Lot of branching will be present. So alpha 1,6 will also be there. Okay, so this is all about homopolysaccharides. Now we will discuss about heteropolysaccharides, and also a small note on proteoglycans. Okay. Okay, so to start with heteropolysaccharides. Heteropolysaccharides are actually, the main heteropolysaccharide we see is mucopolysaccharide. Okay, mucopolysaccharides, which is otherwise known as glycosaminoglycans. Glycosaminoglycans. Okay, or usually abbreviated and said like GAGs, glycosaminoglycans. Okay, this is the main one that we have to learn. And the other three that are present are, one is agar, another one is agarose, and the fourth one is pectin. Okay, so I think we'll cover the discussion of this first and then go to the main one. Now talking about agar, as you know, you might have already studied about agar. This is a polymer of, now before coming to this, just as an intro to heteropolysaccharides, these are repeating units. Like homopolysaccharides, the repeating units were similar, same units were repeating, but here different units will be repeating. So usually these are different units repeating of different units, repeating units of, or repeating different units, or monosaccharides. Okay, different units, that is known as heteropolysaccharide. Now coming to agarose. Agarose is a repeating unit of galactose and glucose. Galactose and glucose polymer. Okay, it is a polymer of galactose, glucose like that. So that is known as agar, and agar is usually used as a medium, agar medium. You will study later in next year in microbiology that it is used as a culture medium for bacteria. culture medium in microbiology for bacteria. Okay, so this is the main application of agar. Whereas agarose, when you come to agarose, it is a polymer of galactose and N-hydrogalactopyranose. Okay, N-hydrogalactopyranose. Okay, galactose and N-hydrogalactopyranose. Now, in agarose, what happens? Agarose is usually used as in electrophoresis. You will again study in your later classes or next year when you go to not this year itself, you might have studied about the techniques, right? So electrophoresis is one such technique. So for protein, DNA, RNA electrophoresis of any of these protein, DNA, RNA or any nucleic acid, we can use what? Agarose as a gel medium or the medium for electrophoresis. So such electrophoresis is known as agarose gel electrophoresis. Okay, agarose gel electrophoresis. So agarose has such an application. Now pectin is a combination of galacto, I think galactouronic acid. Yeah, the main content is galactouronic acid. Okay, galactouronic acid. Yes, galactouronic acid is the main content. Some other monosaccharides are also there in repeating units, but the main content is galactouronic acid. Now pectin is used as a gelling medium in food. Okay, creating jellies, like you have some kind of edible jellies, right? So as a gelling agent in food also pectin is used, and this is also a important dietary fiber. Okay, dietary fiber. So compare and study the just before this discussion we had discussed about homopolysaccharides. Their cellulose and inulin were dietary fibers. The dietary fiber which comes under heteropolysaccharide is pectin. Okay, so this is about agarose and pectin in short. So we'll focus our discussion to mucopolysaccharides or GAGs. Okay, I think in my last class towards the end when I told about heteropolysaccharide, I told mucopolysaccharides are the synonym of heteropolysaccharide. That is wrong. Heteropolysaccharides are many, 1, 2, 3, 4. These all are heteropolysaccharide. The first one is this. Okay, must be a slip of tongue, sorry. So we'll focus our discussion on mucopolysaccharides. Okay, so mucopolysaccharides. Now in mucopolysaccharides we have 6 main mucopolysaccharides or GAGs. So we'll study the composition of each glycosaminoglycan, where it is present in the body and what are the applications of that being present in that area. So, and also there are some diseases associated with these which are known as mucopolysaccharidosis. Okay, MPS. So you don't have to know that in detail, but I'll share the details of 1 or 2 or 3 main mucopolysaccharidosis in the group. Okay, all these are actually being asked as a MCQ, not for theory. Main questions and all won't come from this, but still as an MCQ you have to learn all this. Okay, so mucopolysaccharidosis, if you discuss about mucopolysaccharidosis or GAGs, what are these? Now mucopolysaccharidosis are unbranched, okay, these are not branched, these are linear polymers. Unbranched polysaccharides are heteropolysaccharides composed of heteropolysaccharides composed of disaccharides. Okay, now what are these disaccharides, or the two units which you call as disaccharides? Disaccharide means two units should be there. So one unit is this, one unit is this. What is the first unit? Is a amino sugar. The other unit is a uronic acid. Okay, amino sugar and uronic acid. Now what are the amino sugars that can be present here? One is glucosamine, N-acetylglucosamine, or simply as glucosamine can also come. Glucosamine and galactosamine. Any of these can be present. Glucosamine and galactosamine. Okay. Uronic acid we usually see are D-glucuronic acid. Glucuronic acid. And the second one is L-iduronic acid. Okay. L-iduronic acid. Okay, so every mucopolysaccharide will the body when it wants to create a mucopolysaccharide, it will take one amino sugar and one uronic acid and mix it to form a mucopolysaccharide. So we'll learn the list of the 6 mucopolysaccharides, where in body they are found, and what is the function of that molecule in that area. Okay, yes. So for that I will be needing the whole board. Okay. Now here we will discuss the 6 main heteropolysaccharides or the mucopolysaccharides, the areas they are found, and the functions of these molecules in those areas. Now one or two diseases, the diseases associated with these mucopolysaccharides are known as mucopolysaccharidosis. Okay, MPS. So there are actually 6 or 7 mucopolysaccharidoses. You don't have to learn at your level, you just understand there are maybe 1 or 3 main mucopolysaccharidoses to study. I'll share those details in your group. Okay, only that you study, and the enzyme deficiency. Sometimes it can be asked for MCQ. Okay, not for the main question. So we'll start with mucopolysaccharides. So in order to remember these mucopolysaccharides, we will create a table here where you can match each other, one component from here and one component from there, and create each one. Okay, so it will be easy to remember also. So the first component is an amino sugar. I told you one amino sugar should be there, right? And also a uronic acid should also be there. Yes, uronic acid. What are the amino sugars? One is glucosamine. Another one is galactosamine. Yes. The uronic acids are D-glucuronic acid and L-iduronic acid, right? Okay. So with these in hand, we will start discussing about the mucopolysaccharides. Now the main mucopolysaccharides that we are going to discuss are hyaluronic acid. You might be familiar with this hyaluronic acid from anatomy or histology. Next one is chondroitin sulfate. The third is dermatan sulfate. As the name indicates, this is mostly widely present in the skin. Dermatan sulfate. And the next one is heparin, which is an anticoagulant that you might be already familiar with from physiology. Heparan sulfate is the fifth one, heparan sulfate. And the final one is keratan sulfate. Okay, keratan sulfate. Now we'll learn the composition of each, where they are distributed, and the main function of these things. Okay, so mucopolysaccharides, we'll create a table on this part. So first we'll see the mucopolysaccharide here. So the first mucopolysaccharide is hyaluronic acid. Okay, we'll denote HA, just to you to understand. Okay, HA is the hyaluronic acid. Next one is chondroitin sulfate. Next one is dermatan sulfate. Next one is heparin. Next one is heparin sulfate, and keratan sulfate. Okay. Yes. So coming to hyaluronic acid. So what is the composition of hyaluronic acid? Now hyaluronic acid can be combined. The composition will be N-acetyl glucosamine and D-glucuronic acid. Okay, the first pair, N-acetyl glucosamine and D-glucuronic acid, will give you hyaluronic acid. Okay. Yes. Also, one code that I usually remember is HCD and HHK. Okay, the mucopolysaccharides, if they ask, this is the list: hyaluronic acid, chondroitin sulfate, dermatan sulfate, heparin sulfate, and keratan sulfate. Okay. Yes. Now talking about the constituents. Hyaluronic acid, as I told you, N-acetyl glucosamine and D-glucuronic acid, that are the components. N-acetyl glucosamine, I am not writing full, N-acetyl glucosamine, and the first pair, which is the D-glucuronic acid. Okay, D-glucuronic acid, that is the first pair. So we'll see all the compositions first and then discuss the distribution. So that is the first, hyaluronic acid. And coming to chondroitin sulfate, the combination is N-acetyl galactosamine and D-glucuronic acid. The first one was N-acetyl glucosamine and D-glucuronic acid. Chondroitin sulfate is N-acetyl galactosamine and D-glucuronic acid. Okay, N-acetyl galactosamine, I am not writing full, N-acetyl galactosamine and D-glucuronic acid. Okay. Maybe we'll create some space here. So we have dermatan sulfate next. Okay, and heparin sulfate and keratan sulfate. Okay, so next is dermatan sulfate. Dermatan sulfate, the combination will be N-acetyl galactosamine and so next is dermatan sulfate. Dermatan sulfate, the combination will be N-acetyl galactosamine and L-iduronic acid. Okay. Plus N-acetyl galactosamine and L-iduronic acid. Galactosamine and okay. Now the next heparin combination will be glucosamine, chumma glucosamine, no N-acetyl attached, it is glucosamine plus iduronic acid. N-acetyl chumma glucosamine and iduronic acid will give you heparin. Now heparin sulfate is glucosamine and the other one, glucuronic acid. Okay, understand the difference, easy to remember. This is glucosamine and hydronic acid. This is glucosamine and glucuronic acid, Hs. Now keratin sulfate is simply N-acetyl glucosamine and along with that galactose. Okay, chumma galactose, no attachment, no derivative, it is just galactose. So these are the main 6 mucopolysaccharides that you have to learn. Okay, please understand, the first one is hyaluronic acid. Hyaluronic acid, we take N-acetyl glucosamine and D-glucuronic acid. For N-acetyl glucosamine and D-glucuronic acid. For chondroitin sulfate, we take N-acetyl galactosamine and D-glucuronic acid. Okay, for hyaluronic acid, it is N-acetyl glucosamine and D-glucuronic acid, whereas for chondroitin sulfate, it is N-acetyl galactosamine and D-glucuronic acid. Okay, now coming to dermatan sulfate, we come to the next thing, which is N-acetyl galactosamine and L-iduronic acid. Okay, L-iduronic acid. Now what next is heparin. Heparin and heparin sulfate is easy to remember. Both has glucosamine, but with it the attached acid is different, right? Heparin has iduronic acid with it. Heparin sulfate has glucuronic acid with it. So the final molecule is keratin sulfate, and keratin sulfate is easy to remember. You take the first one, N-acetyl glucosamine, and together with that you have simply galactose attached. Okay, simply galactose attached. So these are the main mucopolysaccharides present. Now the areas present of each is easy to remember actually. Hyaluronic acid are found in synovial fluid. Okay, I think you have studied this in anatomy histology. Synovial fluidum and also in vitreous humor of the eye. Okay, vitreous humor of the eye. Okay, vitreous humor of the eye. Now chondroitin sulfate is usually found in cartilage, bone and tendon. Okay, cartilage, chondroitin. Again, you might have learned in osteology, the basics of osteology. Chondroitin will be a term that you will be familiar with. Cartilage, bone and tendon, it is predominant. Okay, bone, tendon and cartilage. Now coming to dermatan sulfate, I told you it is the main thing present in the skin and also heart valves. If you want to remember this also, heart valves also it is present. And heparin as such is found, yes, lungs, mast cells, yes, mast cells, lungs, skin and all. Okay, but the main thing in skin is, so I think you remember these two. Mainly in lungs and mast cells you have heparin. Okay. So heparin sulfate is, maybe you can remember skin also with it, lungs, mast cells and skin, whereas heparin sulfate is skin and basement, kidneys, basement membrane. Okay, I hope you remember what is the basement membrane of the kidney. Might be covered detail in your physiology, nephrology classes, right? So you have skin and basement membrane of the kidney. Okay, this is kidneys, basement membrane. That is heparin sulfate. Now keratin sulfate is easy from the name itself. Keratin you can imagine where it will be present mostly in the cornea, right? Keratoconus is the bulging of the cornea. So kerato means mostly mentioning to cornea, and also is present in the cartilage. Okay, I hope it is clear. Hyaluronic acid is found in synovial fluid and vitreous humor of the eye. Chondroitin sulfate in the cartilage and bone, tendon. Dermatan sulfate in skin and heart valves. Heparin in lungs, mast cells and skin. Mast cells are involved in allergy, which release histamine and all. So heparin sulfate is found in skin and kidney basement membrane. Okay, keratin sulfate in cornea and cartilage. So these are the main mucopolysaccharides that we have learned. Hyaluronic acid, we learned how to build each mucopolysaccharide, and each mucopolysaccharide, where are they seen. Okay, hyaluronic acid is seen in the synovial fluid and vitreous humor. This chondroitin sulfate is a part of cartilage, bone and tendon. Dermatan sulfate is a part of skin and heart valves. Heparin and heparin sulfate together, you can remember, mostly in the skin, lungs and mast cells, but heparin sulfate basically, the important thing is kidney's basement membrane. Keratin sulfate is found in the cornea and cartilage. Okay, so now there are applications of each. What is the use of each of these mucopolysaccharides in each of these areas? I think I'll share the table in a table format, easy to understand, nothing new. So you can learn from that. And also there is a small topic, nothing much to discuss, about proteoglycan and glycoproteins, and there is a small diagram about proteoglycan aggregate. So I'll share that diagram in your group. Okay. So main things are these. You have to understand the list. You should learn the list of mucopolysaccharides and 2 or 3 mucopolysaccharidoses, again, which I'll share in the group. So hope this was useful. We'll meet in the next class, which is about carbohydrate metabolism, first class. Okay. Thank you.